Embryo. Tell us how the muscle begins to develop in the embryo.
Well, there are basically two. Well, as I described, I mean, there are two groups, two family of genes that are critical for determining the fate. The first one being the PAX, two members of PAX family of transcription factors, PAX3 and 7. And PAX3 actually is the first one to be in action, and it's going to not to lead the cells to become myogenic, but to maintain them in a state where they will be able to respond to myogenic clues in the next steps. And these PAX3 and 7 genes, therefore, are seen as the first transcription factors that are going to lead to the appearance of the progenitor cells that will be at the origin of all the muscle of the limbs, as I said, and the trunk. We did not talk about the head. Head muscles, it's a different story. It's not a completely different stories, but PAX3 is not in action for these muscles. So specification, PAX3, PAX7. And then determination, differentiation, the second group of genes, the muscle regulatory factors, MIF5, MRF4, MyoD, followed by myogenin. Myogenin is a close member of the family, but myogenin, as I said, is the one that is going to push the cells to the terminal differentiation, which means the formation of the fibers. So the first fibers forming in the embryo, and the rest of the life of the animal, even in the adult, require myogenin. So that is the say, very quickly and simply summarized, the beginning of myogenesis in the embryo. Then comes the question of the adult, as you mentioned, the adult. But the question of the adults, actually we see the same players, the PAX3 and 7, but now say PAX7 is before in some way PAX3, because PAX7 is going to be expressed in all the cells that are seen as the satellite cells. Or satellite cells is an interesting name. As I said, it comes from the definition given by Alexander Mauro, who was a cytologist and histologist, and who was observing muscles and found these few cells at the periphery of the fibers. Periphery, satellite, simple. But for these cells to be maintained and to be active all along the life of the adult, the PAX7 gene is required. So loss of PAX7, loss of myogenic activity in these cells. And loss of these cells is loss of the regeneration potential. So that's absolutely essential that the PAX7 gene be maintained, expressed in these cells. This will permit, by a mechanism that we see as the control of quiescence, because these cells, in absence of muscle overuse or muscle injury, the satellite cells behave exactly as, say, skeletal muscle stem cells. They remain next to the fibers, sleeping, quiescent, but ready to wake up and to repair or regenerate the injured fibers or the missing fibers. And again, the second family of genes, the MRF, muscle regulatory factors like MyoD, MIF5, and so on, are also absolutely necessary but for the step that follows, the maintenance of the say specification state, that is remain ready, remain dormant, but ready to activate the program in order to be able to repair the muscles when necessary. The activation of these cells seems to be affected in many situations like this one, and therefore they cannot do their job properly. And there are experimental models that are being worked for trying to understand more about the signaling and how we could intervene on this type of signaling so that the cells could ignore or we could attenuate simply the signals or suppress them that prevent these adult satellite cells or skeletal muscle stem cells prevent from undergoing activation and differentiation and reconstruct missing fibers or altered fibers. That is a very interesting. The control of quiescence is extremely important because we've seen that we've seen that altering quiescence is possible in vivo in the physiological condition, even if an injury is a physiological condition. And the cells, most of the cells that are mobilized, activated, so the progeny of these satellite cells will differentiate, form new fibers, and a subset of them will return to quiescence, provided that the signaling that is required for this is not affected. And as I've shown, the notch discussed. This is the work of other labs. The notch signaling is absolutely required for maintaining quiescence. You disrupt the notch signaling, satellite cells undergo activation, aberrant differentiation, and the reservoir is empty after a few weeks or a few months, depending. So that is also a very important aspect of the physiology of muscle regeneration. Make sure that self-renewal and quiescence will be maintained. I have to think there is a lot to do still in the field of myogenesis, developmental and adult. I would say that our work was really mixing together. It's the conjunction of the developmental myogenesis and regenerative myogenesis, and by mixing the two, we've been able to say probably progress faster than we thought because of observations that came from the developmental field and that helped us, like the PAX3 knock-in. I mean, initially this knock-in was produced in the lab to track the early steps of myogenesis. It was not at all prepared or made initially to track the satellite cells in the adults. And the big surprise was that it turned out to be super useful for isolating these cells. So, voilà, I would say mix the fields.
Excellent. Thank you.
So atrophy is a severe problem that happens in patients who have cancer and COPD, chronic obstructive pulmonary disease, and other serious diseases, and it causes a condition. Those diseases cause a condition called cachexia, which is the loss of skeletal muscle that cannot be reversed nutritionally, and you get this very significant atrophy or loss of skeletal muscle, even if you're getting parenteral nutrition like amino acids, this kind of thing. So you're in a negative nitrogen balance, and that causes an increased risk of death from these already serious diseases. Yeah, so the atrophy is read out by a loss of mass, and of course that causes profound weakness, so that you can't easily climb a flight of stairs, you can't go about your normal life. And this is also caused by other conditions like the loss of muscle with old age, which is called sarcopenia. I'm sure people are familiar with the frailty of the elderly, and the muscle component of that is sarcopenia, and the bone component, as everyone knows, is osteoporosis. Right, so one of the major mechanisms that's a negative regulator of muscle mass is a series of molecules in this family called the TGF-beta family, and these include myostatin, activins, and GDF11. These are all TGF-beta like molecules, and they go through a receptor called the activin type 2 receptor. And they've all been shown to block muscle differentiation and block muscle growth. And they inhibit, downstream of this receptor, a protein that's required for protein synthesis, or actually not necessarily required, but is able to stimulate protein synthesis, called AKT. So, for example, when myostatin stimulates the activin receptor, that blocks AKT, and therefore it blocks protein synthesis. So our strategy is to inhibit the activation of that activin receptor, and therefore block these negative regulators of skeletal muscle mass, and therefore restore muscle size in disease settings. You would want to show that in a setting of atrophy, for example, if you have a cancer cachexia, like a tumor which causes the significant cachexia and atrophy, if blockade of the activin receptor is sufficient to restore the muscle to normal size. A study like that was done by another group, and it was really interesting because the tumors as you can imagine, can cause death of the animal, but in the setting of a inhibitor of cachexia that did not touch the tumor, these animals lived significantly longer. So it really showed that you could just block the cachexia and cause an increase in survival, which is quite a surprising result. There's an increasing number of people who are living longer and longer. The percentage of people who are going to be above the age of 65 will be about a third by the time 2050 comes around. And so there'll be many more people who will be at risk for sarcopenia and frailty. And the real hope is to allow people to go about their normal lives and not end up in old age homes because they can't get up a flight of stairs, or they can't get out of bed or out of a chair. So these, people may not focus on how serious these problems are, but sarcopenia is a reason people end up in an old age home, for example, or assisted living situation, because they can no longer have the strength to take care of themselves. So the hope is, that this will treat these people, or these kind of mechanisms.
I think you said something like muscle is the last untreated target organ, or something of that sort.
Yeah, it's surprising because muscle is about 40% of your body, and there's very little that's available to patients currently. I mean, in some settings, there are low-dose glucocorticoids, or glucocorticoids are used in some settings, but it is a relatively untreated organ, which is strange if you think about it. So hopefully we can do something about that. We're beginning to understand the molecular mechanisms that really underlie loss of muscle in serious disease, and it does seem to be possible to block those mechanisms or stimulate positive mechanisms, and therefore it should really be possible to treat serious human disease where there's a significant loss of muscle mass, which can result in early death and loss of mobility and loss of function. So I think it's good news that it seems to be possible to do something about that.
Yeah, I think if I may kick off this one, that was a very important session, especially as muscle and bone. We know more and more that they go together. If you lose muscle mass, it has implications for your bone. But I think Didier Montaras introduced the bone field, to a very special cell, the satellite cell. He did it from a very developmental perspective, but very clearly. And it showed us that these are the stem cells of the muscle that can make muscle cells. And he showed some of the key regulatory components in there, like notch signaling, and especially PAC7. That was for me very important to hear. But
For me, it's interesting. I mean, like for the osteocyte, which was there around, most frequent cell, but nobody really understood the function, had the means, the technologies, the methods to study this. It appears that this concept of the satellite cells that has been around at least since the 60s, now is really being well understood, or much better understood as this reservoir of the muscle stem cells. And he in my view, presented very elegant experiments where he specifically impaired this reservoir and looked at the consequences on muscle regeneration. And one example was that he used tamoxifen to basically induce the damage, and previous studies over 1 or 2 months didn't see anything, but okay, being a good researcher, he looked on, and after 5, 6, 8 months, indeed there was the impairment that would have been predicted.
which might, in hindsight, be logical. I think that you don't waste your muscle. You only need to repair them, and that takes a long run. It looks like bone research, that you have to be patient to see changes at the bone level. And but it was very instructive for us. I think the people in the bone field not working on muscle, and the approach, and also the genetic tools to dissect the function, and what he presented about the critical role of PAX7, that should go up in the satellite cell to be able to become in an activated state, and the notch signaling critical to have the satellite cell in a quiescent state. So David Glass in the next presentation, who was less at the molecular level but more on the concept, how can you develop therapies to stimulate satellite cells to repair muscle or to regain muscle mass. First of all, it's very nicely showed that also understanding the molecular mechanism, how satellite cells can be differentiated, is important, but also documenting. And that was, for me, I didn't know that the first sign of sarcopenia is the decrease in mitochondria in the muscle cells. That is really a sign, a very early sign of change in muscle function. Was for me very, very important to hear.
Yeah, he gave a very nice introduction, sort of taking it from the previous field and bringing out the two main facets of muscle atrophy, one in the setting of cachexia and then sarcopenia, both being entities that need to be treated. And it was very interesting that he said there is virtually no specific drug for this area. It's one of the few areas where there is not much that physicians have at hand, aside of glucocorticoids or others that of course have many fields. Yes.
Yeah.
So definitely.
Were you aware of that?
No. I mean, I thought muscle is important, so probably many companies would have thought about bringing something forward, but maybe we now just have the means and understanding to do this. And
that's what they are working on now, understanding the mechanism and looking at experiments of nature. The Belgium cow and the dog, but also the little boy now identified that if there is something wrong in the myostatin signaling cascade, which is an inhibitor of muscle cell development, that you really can have massive muscles, like the German cyclist pictures he showed. It was really impressive, and but also it shows that you have a new target for trying to intervene, and I think they are, at least what he showed, on the way to intervene in that mechanism to see whether that can be used for sarcopenia. Yeah, I mean,
it seems like a common theme is inhibit the inhibitor. That seems to be, in many cases, the doable way of
the parallel with the osteoblast inhibitor. That's true. And what the interesting thing is that if you look, it's the signaling cascade of that myostatin really overlaps with the TGF-beta, or more the BMP signaling and the activin signaling, And those signaling cascades are also very important in osteoblast development
Yeah.
and osteoblast function.
Yeah.
So he didn't touch upon that, but I think there are compounds that can do both,
Yeah.
that have an effect, positive effect on the bone, cell development and bone formation, and have an effect on the muscle mass. So I'm really curious to see what will happen in that field in the near future.
And that's obviously also very important development right now, that again, like in the bone field, we differentiate between mass and function.
Yeah.
And the strength on the bone as well as on the muscle
Yeah.
obviously is more important than just having more mass there.
Exactly. And
And,
how does the mass look like?
Yeah.
Bone matrix, how does the
Yes.
bone matrix? So there are
Yeah.
really parallels in that.
So we need to learn what are the indicators that best then also show progress due to treatment.
Yeah.
Do we need to look at strength or power or coordination? I mean, there are many facets, I think, to look at muscle function, and that has
Yeah,
become an
absolutely.
increasing topic also in our musculoskeletal setting here.